Okay, you guys, welcome back to hematology. As I may have mentioned before, I really like to recap, and sometimes I will throw together a recap PowerPoint presentation just to kind of bring everything together and go over what we've already gone through. So this is the third lecture. It is basically a recap of red blood cells and hemoglobin. It's a very brief overview, just give you some additional information. It's not a very aesthetically pleasing PowerPoint, but utilize it just to kind of briefly recap everything we've been over. So again, just a reminder, our mature red blood cells are biconcave with a central pallor and they lack a nucleus. Hemoglobin contained in the red cell transports oxygen throughout the body, and the average lifespan for a red blood cell is 120 days. Cells are pliable, aka deformable, so that they can effectively move throughout the body's vasculature. Remember, the two most important characteristics is permeability and deformability for red cells. Erythropoiesis is again that production of red blood cells that occurs in the bone marrow. It encompasses all stages from that hematopoietic stem cell through a mature erythrocyte. And remember, make sure that you know both nomenclatures for each stage of red blood cell development. We have erythropoiesis that takes place in very specific areas called erythroblastic islands. Those are kind of clusters of red blood cell production. And if you look at the picture, they're clustered, these red cell, immature red cells are clustered around this iron-laden macrophage. So remember, resident macrophages of the bone marrow store iron. We call them siderophages. And you can see the little granules of iron inside the vacuoles of this larger cell in the middle, and that is a siderophage or a resident macrophage that is supplying iron to the developing red blood cells, as well as cytokines to aid in maturation. I'm not going to expect you to know the name of the cytokines, but I do want you to know that there are chemical signals happening that promote and instigate maturation and development. The heme pigment of the hemoglobin facilitates oxygen transport to the tissues and CO2 from the tissue. So they bring oxygen and remove waste. Substances required for red blood cells and hemoglobin production. So again, we need the building blocks to efficiently perform erythropoiesis. So amino acids, which are used as building blocks for proteins in our globin chains, iron, B12. Now let's remember B12, vitamin B6, and folic acid and folate are all imperative for DNA synthesis. Cobalt and nickel, which you wouldn't really think about. And to be honest with you, I don't know the exact biochemical mechanisms in which cobalt and nickel play a role, but they are also important in production of hemoglobin. Deficiency of any of these substances can result in an abnormal erythropoietic process. Iron from senescent red blood cells, remember, senescence means aged or breaking down. We call them pycnotic. These red blood cells get recycled by the macrophages and taken back to the bone marrow where they are used, those building blocks are used to build new cells. Erythropoietin is produced primarily by the pertubular cells in the kidneys. They have sensors that help regulate EPO production or erythropoietin. So when we have a partial O2, so it has a sensor for partial O2 levels, and when we have a decreased level of O2 circulating, those sensors will cause an increase in the erythropoietin production. A small amount of erythropoietin is produced also in the liver, but the primary location for adults is in the kidney. And remember, during fetal development, we have erythropoietin production primarily in the liver during fetal development, but as a fully formed human being, most of that comes from erythropoietin comes from the kidneys. Once the stem cell differentiates into the erythroid cell line, remember, it branches out. A cell matures through nucleated stages in 4 or 5 days. So from the very beginning, when we have that pronormoblast or that rubroblast, that is day 1, and by the time it is 4 to 5 days, we have moved on to the reticulocyte stage. So bone marrow reticulocytes have an average maturation period of 2 to 5 days, and that's one of the reasons why we kind of see them circulating in the blood more often. But once they are sent out into circulating blood, they remain in the reticulocyte stage for another day or so. Reticulocytes represent approximately 0.5 to 1.5% of the circulating erythrocytes. So again, it's normal to have about a 1% turnover rate because as new cells are coming into circulation, old cells are being taken out. So it's a constant turnover rate. But it's important to know what the normal range of reticulocytes should be, so 0.5 to 1.5. And again, when we are testing or performing a reticulocyte count for analysis, we can only use blood that is less than 24 hours old. And this is why, because over that one period, average 1 day, those reticulocytes mature into fully mature red blood cells and are no longer reticulocytes. So a lot of testing has limited time frames in which we can give them accurate information. Okay, and the brief, you guys have had this information given to you already, but just I thought I would post it alone. So the stages of red blood cell maturation, know both of those nomenclatures. Pronormoblast, which is also called a rubroblast, basophilic normoblast, aka the prorubrocyte, polychromatophilic normoblast, the rubrocyte, orthochromic normoblast, the metarubrocyte, polychromatophilic erythrocyte, also known as the reticulocyte, and then last but not least, we have the mature red blood cell, aka the mature erythrocyte. So remember, for your exam, you're definitely going to need to know both of those nomenclatures, but you're also going to need to potentially know it for your board exams with the ASCP. Now, we really haven't gone into much detail about each one of these phases of development for the red blood cell, but let's talk a little bit about them now. So we are going to go over in class during our student lab activity. We are going to look at our cell atlas and also some bone marrow slides, but just so that you have a visual reference in addition to the in student lab time, let's talk about the characteristics of each phase of development. So the rubroblast, also known as the pronormoblast, is typically the largest of all of the developmental phases. We have a nuclear to cytoplasmic ratio of 4 to 1, meaning we have 4 times the amount of volume of nucleus as we do to cytoplasm. Typically, the size of these rubroblasts are 12 to 19 micrometers in diameter, and of course, we've got a large round nucleus, chromatin that has a fine pattern, which means it's smooth looking. If you look at this chromatin compared to, let me get my pointer so you can see. So here we have our rubroblast. If you look at the chromatin here, it looks nice and smooth, homogeneous, but here you've got areas of clumping. So the more mature the cell is, the more densely clumped or densely compressed the chromatin will be in the nucleus, and that goes for both red cells that are developing and white cells. So smooth chromatin is going to be a definite sign of immaturity in all of the immature cells that we see. Okay, you might see 0 to 2 nucleoli. I don't know if you can see this well, but these tiny little dimples, this one doesn't have very prominent nucleoli. But any type of blast, whether it's a white blood cell or a red blood cell blast, um, whether it's a myeloblast or a rubriblast, are prone to have areas of clearing called nucleoli within the smooth chromatin of the blast phase. The cytoplasm is a distinctive basophilic color without granules, okay? So we don't see any granularity here in the cytoplasm, but it does have that just that bluish purple color or that pinkish purple color all the way around, okay? The next phase of development is the prorubrocyte or the basophilic normoblast. We're going to see the diameter in this cell shrink just a little bit from 12 to 14 micrometers in diameter. Again, our nuclear to cytoplasmic ratio is still going to be 4:1, but we're going to start to see more clumping in the patterns of the nuclear chromatin. So it's going to look kind of like darker in some areas, kind of clumpy. I always try to describe it like when you get a cone of cotton candy, it's nice and fluffy and everything is kind of like, consistently fluffy. But then as you squish it down into a ball or you squeeze it or you pull some off, you start to see it kind of condense and clump together. That is kind of how I imagine chromatin within nucleus to be. They just kind of like as they get older or more mature, they start to clump. And then we've got a distinctive basophilic color as well. And you can see it's a little bit less, it's a little bit less, I guess it's like a little bit more basophilic than the blast was, if that makes sense. So if you compare the picture prior to this one, you can see the difference in the cytoplasm. All right, and even more clumping now. So do you see what I mean about the clumping? It's more condensed. Okay, so again, we are shrinking in size. We're looking now at a polychromatophilic normoblast, also known as a rubrocyte. The size is going to be a little bit smaller at 11 to 15 micrometers in diameter. And we're going to have a nuclear to cytoplasmic ratio of 1:1. So nucleus is here, but you're going to have a little bit more cytoplasm. So you're going to have pretty equal parts cytoplasm to nuclear material. The nucleus is going to demonstrate much more clumping of the chromatin, and you can see that. Those kind of looks chunky. You have little areas called parachromatin where you have clearing in between the clumped chromatin. And then we're going to have a variable colored cytoplasm with pink staining mixed with basophilia, so some light and some dark areas. All right, and then we have the metarubricite. So this is the phase of these are most common nucleated red cells that we see in this phase. They are much smaller. They are 8 to 12 micrometers. And we've got very, very condensed chromatin now. You don't have any of that parachromatin. It looks very condensed and tightly packed into this little tiny pellet. And you'll notice in the metarubricite, or also known as the orthochromatophilic normoblast, that the nucleus is no longer central. It is kind of to the periphery, and it looks like it's going to fall off, and it will be ejected soon. The cytoplasm is going to be acidophilic, which is reddish pink, which is more representative of a typical red blood cell rather than that darker color. Right, so if you look here in the bone marrow, you can kind of see the representation of the different phases. So here we have the prorubrocyte, or prorubrocyte, or so that's the second phase. Then we've got the metarubricite. So see how much smaller it is in comparison. And then here we have the rubrocyte, which is a little bit between the prorubricite and the metarubricite. So we're going to be looking at some of those slides as well. All right. So remember, reticulocytes are about 1, so I would say 0.5 to, I would say 2%, 1.8%. I mean, it's kind of in that range. Reticulocytes, again, they appear as polychromasia. And then here we have a metarubricite, which is also called a nucleated red blood cell when we're reading our slide. To quantitate as a reticulocyte. So if a physician specifically orders a reticulocyte count, we can't base our count on polychromasia. We actually have to go in and stain our slide with new methylene blue. And the reason for that is we have to essentially prove that the cells we're counting have the RNA, residual RNA present. And here, when this bottom picture, this new methylene blue stain, we can definitely tell that these two are very clearly reticulocytes. The reason for that is reticulocytes are increased when the bone marrow is trying to replenish the red blood cells. So if we have acute blood loss, hemolytic anemia, hypoxia, etc., we want to see an increase in reticulocytes. If the patient is anemic, has low or decreased hemoglobin, has had an acute blood loss, and we don't see reticulocytes, that's when we really get concerned, because that means the bone marrow is not compensating for the need. And we have to investigate, is this patient lacking building blocks? Is there some type of malignancy that is overriding red blood cell production? What is happening? Is the patient in kidney failure, so their kidneys are not producing erythropoietin? We have to find out the cause of the reason for the bone marrow not producing more red blood cells, and in this case, we would see fewer reticulocytes. We want to see polychromasia. We want to see reticulocytes, especially when there's an increased need. And we want to see some because it's a normal part of our circulation, just as we replenish, just to indicate that we are replenishing the red cells that have been destroyed as a normal part of red cell senescence, if that makes any sense. Okay, so again, in order to aid and recognize problems, we have to know normal ranges, right? So our reticulocyte count is expressed as a percentage of total erythrocytes. So we have a range of 0.6 to 1.8% in males, and then in females, 0.5 to 1.6%. So let's round it up to 2. So 0.5 to 2. Since it is a percentage of the total red blood cells available, a corrected reticulocyte count should be reported as to not overestimate the retic count in anemic individuals. Because in anemic individuals, they may have a lower red blood cell count overall, a lower hemoglobin, they're going to need to have an adjustment made with using this calculation. So luckily for us, our Sysmex analyzers do this calculation for us on our behalf, but it's very important for you to know how to do it. And the reason for that is if you work in a facility that does not have the LIS is capable of doing that calculation, or if your reticulocyte count exceeds the linearity or the capability of the instrument to accurately count the reticulocytes, we have to do a corrected. So the corrected reticulocyte is going to equal the raw retic percent times your red blood cell count divided by 5. All right, so It's a very easy calculation, but know this calculation and be able to utilize it. And we'll talk more about this in class, but essentially we are going to count 1,000 red cells and we are going to take note of how many of those red cells are reticulocytes, calculate a percentage from that, and then multiply that by our red blood cells divided by 5. And our red blood cells are going to be told to us, going to be reported to us on that Sysmex or that hematology analyzers print out. So unless there's something faulty or it's giving us some type of flag or error in process, we can utilize the automated red blood cell count to plug it into this calculation. So very, very easy. All right. After the reticulocyte stage, remember, once it's a reticulocyte in circulation, it stays that way for about 24 hours. The mature erythrocyte is formed. And of course, these are non-nucleated cells. They transport oxygen to the tissues via hemoglobin. They survive in circulation for approximately 120 days, and they metabolize glucose through anaerobic glycolysis. The average diameter is 6 to 8 micrometers. It lacks the ability to make hemoglobin because remember, hemoglobin has to occur during the nucleated phase of development because it relies on that mitochondria to create that porphyrin or that protoporphyrin and insert it into the heme. It also lacks a nucleus and all functional organelles. But it still does a fantastic job at what it's meant for. All right, so there are some disorders related to erythrocyte maturation and production. I'm going to leave this in here just for you to come back as a resource. However, this is going to be more a part of our white blood cell unit, even though it is associated with red blood cells, because there are some genetic gene mutations that are associated with proliferative disorders, and that kind of overlaps into our leukemias and our myeloproliferative disorders. And remember, red blood cells do come from a myeloid stem cell, and so it is considered a myeloproliferative disorder when it is overstimulated. So therefore, we're going to kind of not go over this in depth, but I'm going to leave it into but I can't talk. I'm going to leave it in this PowerPoint so that you can come back and look at it later on if you need to. Same with this. I'm going to leave this. We can come back to it and discuss it a little bit more once we've gone over those proliferative disorders in our second unit of white blood cells. One of the things that we can see, if you recall, when we talked about our macrocytic megaloblastic maturation or anemia, it's a defect in maturation due to B12 or folate deficiency, and that can be seen in those macrocytic anemias, primarily because B12 and folate play such an important role in DNA synthesis. And when we think about the importance of DNA production and synthesis during cellular maturation and division, if we have all of the building blocks to produce or prepare a cell for division, we might have an expansion of the cytoplasm. And then when it gets time for the nucleus, which has all of that genetic or that DNA in it, it will get to a point where it can't produce enough DNA or can't synthesize enough DNA or ineffectively, and therefore it won't divide. And so you aren't able to create a division of nucleus, but you've already created an enlargement of the overall cell specific to the cytoplasm, and therefore we will see nuclear to cytoplasmic asynchrony in these developing red blood cells because that nuclear maturation lags behind the cytoplasmic maturation, and then sometimes division is not possible. These developing red blood cells may appear younger than they actually are, because remember, in terms of cellular development, the larger cells are typically the younger cells. But we'll talk more about this in our cellular or our anemia unit. It'll be a fun time. Right, so remember, RBC metabolism, these energy resources for the red blood cells are exclusively produced by anaerobic glycolysis utilizing glucose. The main pathway is that Embden-Meyerhof pathway, which is where about 90% of the ATP is produced for the function of the red blood cell, because remember, they are anuclear. We also have that hexose monophosphate shunt, that methemoglobin reductase pathway, and the Lou-Behring Rappaport shunt. We're going to get into more information of those as well. All right, so let's take a look at each one of the red blood cell metabolisms. So again, first off, we have that Embden-Meyerhof pathway, which is that anaerobic glycolysis to produce ATP. We have that hexose monophosphate shunt. And remember, this protects cells from oxidative damage. When oxidative damage occurs, we find that we have an inclusion in the red cells called Heinz bodies because of that denaturation of the hemoglobin within the red cells. And then methemoglobin reductase pathway is basically where that methemoglobin iron that's in the ferric state or the 3+ state it protects us from having that occur. Instead, we need that ferrous 2+ state. So if something goes awry, basically it prevents oxidation of the heme iron. If something goes awry in that methemoglobin reductase pathway, we're not going to have the normal ferrous state. We will have the ferric state, which is harder for our body to utilize and less efficient at doing its job. So the last is the Lou-Behring Rappaport shunt, and remember, that is the part of the red blood cell metabolism that produces that 2,3-BPG so that we can release oxygen or hold on to it based on the need. And we're going to talk a little bit more about this hexose monophosphate shunt coming up with regard to enzyme deficiencies and things that can go wrong there. We know that if the methemoglobin reductase pathway is disrupted, that methemoglobin is a reaction of that, and we know that with the Lou-Behring Rappaport shunt, the 2,3-BPG for cellular respiration would be depleted. So we know what would happen, but we are going to talk more about the hexose monophosphate shunt in just a bit. Here's just a little diagram and a recap for the energy and the ATP production of maintaining hemoglobin function, membrane integrity and deformability, and maintain that adequate supply of reduced pyridine nucleotides. So again, we've got glycolysis, Rappaport Lou-Behring shunt, the glutathione pathway, the pentose or hexose is the same, hexose monophosphate shunt pathway, and here is a reduction reaction that must occur in order for everything here in the the hexose monophosphate shunt or the pentose phosphate pathway, it's the same thing, to occur. And then over here we have another reduction reaction that influences that methemoglobin. All right, looking back at our red blood cell membrane, mature red blood cell membrane characteristics, we have the shape of the erythrocyte, which constantly changes as it moves through the circulation, which is why that cell membrane needs to be deformable. It is composed of a lipid bilayer interspersed with cholesterol and glycolipids. And the cell membrane, again, is deformable and tolerant against mechanical stress and various pH and salt concentrations. So it has to stand up to fluid force in vivo and in vitro. And then ATP is also required to maintain that cell deformability, because loss of that ATP or excess membrane calcium can cause RBCs to become more rigid. Because remember, those calcium ions, they are transported across the membrane through facilitative transport, which means those pumps have to utilize ATP to function properly. If ATP is lost or inadequate, we'll just have excess calcium that can't be pumped out appropriately. And that will cause stiffness and rigidity to occur in that cellular membrane. All right, red blood cell membrane proteins to take note of, and just to review and recap. Remember, glycophorin is that integral protein that comprises the majority of the membrane, so this is a structural protein. We have the sialic acid, which gives our red blood cell its net negative charge, and this is imperative to prevent red blood cell agglutination. Not so much clotting. I think, I probably said clotting before, but clotting actually involves fibrin. But this lack of sialic acid would cause agglutination because those red blood cells would kind of cling to each other, not so much due to fibrin formation, but because they are not propelling one another away from each other as they should due to that loss of sialic acid. Spectrin helps keep that skeletal network of the inner surface of the membrane to keep the cells in that biconcave and deformable shape. And remember, spectrin is going to be really important. If we have a lack of spectrin, we're going to see morphologies that are more susceptible to rupture, like spherocytes. We're also going to have a less efficient oxygen transport system due to a decreased surface area. If proper membrane integrity is not maintained, there will be a decrease in survival time as well, because of the abnormal morphology that it would cause, or the lack of deformability or permeability. And those are recognized by the spleen, or they get caught in the splenic cords because they aren't deformable, or they can't pass through those narrow passageways, and the splenic macrophages will come in, pluck them out, break them down, and transport them or send them to be recycled. Target cells, again, are formed when cholesterol accumulates in the membrane. So that is why they have that bullseye appearance. And then spherocytes and bite cells form when red blood cells are destroyed prematurely. Or, for example, if we have a denaturation of hemoglobin, and we have something like those Heinz body inclusions, they might get temporarily caught up in the splenic cords, and they might have a bite taken out of them by one of those macrophages that are resident there in the spleen. And so it'll just look like, we call them bite cells, because it literally looks like somebody took a bite out of those cells and removed that Heinz body from the cell. Okay, so I don't expect you to memorize all of these. I just want to give you an example of some of the transporter membrane proteins. It's just FYI, we've got a water transporter within the membrane called aquaporin 1. We have an anion transporter, which supports the surface antigens of the ABH or ABO blood type antigens. And then we've got different transporters for calcium. And these are going to be associated with Duffy blood group antigens. So it's really interesting, because the makeup of our inherited protein profile are going to be in very specific variations. Our glycophorin A, our transports negatively charged sialic acids. It supports MN blood groups, and you're going to learn all about these different antigens when you get to your immunohematology blood bank unit or course. But it's really fascinating to know that all of our laboratory analysis or our different areas of study are interwoven. So we really have to learn a little bit of everything as we go along. Glycophorin B and glycophorin C are also part of those blood group antigens, but um, they also perform a job. So they're transporters across the membrane. ICAM4 is an integrin adhesion protein, KEL blood group antigens. So we've got zinc binding endopeptidase supports that KEL group. KID blood group antigens are urea transporters, so that helps get rid of nitrogenous waste across the barrier of our cells. We've got RH blood group antigens, which supports D, C, and E blood group antigens, and then we have RH antigen expression protein. So this is also a gas transporter and probably CO2. Now, I can't pretend to know everything about every single red blood cell antigen. And in fact, a lot of those things are still being investigated and researched. So these are the things that we do know. We know they are transporter membranes. We know some of their functions, but we don't know everything. So keep these in mind moving forward when you get to your immunohematology course. I wanted to present them to you as a little introductory. Okay, so let's recap that red blood cell membrane. Along with the deformability, it is imperative that the red blood cell is permeable. Now, we know it must be freely permeable to water and anions, meaning bicarbonate and chloride. We're looking for a buffering system in our blood pH. We have a very, very narrow window of acceptable pH that is compatible with life. And if we don't have the free permeability to H2O and then anions such as bicarbonate and chloride, we are going to run into problems. It must also be impermeable to cations. So remember, cations are going to have to be selectively allowed in and out of the cell membrane in very specific ratios, okay? So for example, we have very specific intracellular to extracellular ratios for sodium. For every one sodium atom or molecule that is inside of the cell, we have 12 externally or extracellularly. And then the opposite is true for potassium. We have much, much higher ratio of potassium intracellularly as we do extracellularly. So our ratio for potassium is 25 to 1, meaning 25 intracellular parts to 1 part extracellularly. All right. So moving on from red blood cell membrane, let's talk about hemoglobin. Hemoglobin is inherited by our parents. The amino acid sequence that makes our hemoglobin is inherited. Most normal adult hemoglobin is hemoglobin A. It is inherited in a simple Mendelian fashion. And the genotype for normal hemoglobin is AA, meaning you get hemoglobin A gene from one parent and a hemoglobin A gene from another. There are, however, approximately 350 variant types, and that's because hemoglobin defects are mostly due to either amino acid substitutions, which we call hemoglobinopathies, something like a sickle cell anemia, or diminished production of the polypeptide chains. So there might be a deletion rather than an amino acid substitution. We may just have amino acid deletions, and these are called thalassemias. So that is the difference between hemoglobinopathy and thalassemia. Hemoglobinopathies are considered qualitative defects of hemoglobin, and thalassemias are considered to be quantitative. So the first one, there's something wrong with the quality, And the second, there's something wrong with the quantity, deletions versus substitutions. So think of it as you order something from Temu and you get it and you're like, this is not at all what I ordered. It looks like it, but it's not what I ordered. So that would be considered poor quality. Not to offend anyone who loves Temu, I'm just saying. So that would be an example of an amino acid substitution or hemoglobinopathy. They swapped out those cool, that cool skirt you liked or those cool shoes you liked, and you got something that kind of looked like them, but they just weren't what you expected. Or the Temu sent you a package, you open it up and it's empty. You're like, I only have one shoe here, for example. Instead of getting terrible shoes, you get a terrible shoe that's only got one shoe, and that would be considered a deletion, which would be representative of a thalassemia. So that's kind of how I remember it. Qualitative versus quantitative. So again, let's recap hemoglobin structure. We know that normal hemoglobin A is composed of 2 alpha globin chains and 2 beta globin chains. We also know that there are 4 heme groups which are included between those chains, and the heme groups are made up of protoporphyrin rings with an iron molecule there. Now remember, we want the iron to be in the ferrous state, not the ferric state. So that's really important to remember, and we know that our methemoglobin reductase pathway helps us to maintain our iron in the ferrous state. So sometimes hemoglobin has problems because we don't have the building blocks. But remember, it isn't just about supply, it's also about implementation of the iron or the use of the iron. So the very first disorder of iron metabolism would be something like an iron deficiency. So normal iron status continues as iron intake lags. So initially we have enough iron, but then maybe we're not taking in enough in terms of our nutrition, or we have lost a lot of iron because maybe we're going through a growth spurt and we're utilizing it faster than we're intaking it, or we're a woman and we are pregnant and growing another human being, which requires a lot of resources. So eventually, over time, that loss will be too great for the intake to keep up. And at that point, we're going to have a depletion of our iron stores. As those iron stores continue to deplete, we will then have iron deficiency anemia. So it doesn't happen overnight, it happens over time. We see it very often in newborn babies because they have such an increased demand of iron because of their rapid pace of growth. We see it in pubescent children. Again, they have an increased need and they are growing very rapidly. We see it in athletes who are not maintaining proper nutrition to meet the need of their muscle mass. We see it in women of childbearing years due to pregnancy, menstruation, and overall increased need due to those reasons. Again, we also see it in elderly people. So there's a couple of reasons why we see it in elderly people. Number one, the bone marrow becomes less efficient as we get older, and it doesn't produce as well. Also, a lot of older people have poor appetite. They have less less access to nutritious foods because they may be on a very limited budget. There are a lot of things that are involved socioeconomically with people who are older and on a very fixed income. So we have to take all of those things into consideration. The other types of disorders might be a genetic defect of iron. So maybe we have a gene where we don't have like the ability to take the iron that we have stored and implement it into our hemoglobin molecule. We might have overload. So hereditary hemochromatosis is a big one. And then we may have sideroblastic anemia, which is essentially not necessarily an inherited issue, but it is an issue that arises due to an external factor blocking our body's ability to insert iron into our heme groups. So we have plenty of iron in storage, but unfortunately, something like lead poisoning is preventing us from implementing that or inserting that into that protoporphyrin ring of our heme group. Okay, so disorders of heme. So when we think about the production and development of our heme group, we know this is going to take place in the mitochondria during the nucleated stages of development. I'm not going to go too deeply into this, but I do want you to know that porphyrias are a disorder of the synthesis of porphyrin. And porphyrias can be classified based on a few different various characteristics. Number one, clinical presentation, so acute versus chronic. The source of the enzyme deficiency causing the porphyria, and the site of the enzyme deficiency in the heme biosynthetic pathway. And you're going to learn a lot more about this and in greater depth when you get to your enzyme lectures and unit in chemistry, in clinical chemistry. So I'm just introducing it to you now. At this point in time, hematology does come before chemistry, but I think that may change. So this might be a review to you by the time you get to this lecture. All right, our globin chains. So we have a comparative chain composition of hemoglobin types. So remember, at various stages of development, we have different types of hemoglobin based on the phase of development we're in. So we have embryonic hemoglobin, which is we have Gower1, Gower2, and Portland. Gower1 is going to be made up of 2 zeta and 2 epsilon chains. Gower2 is going to be 2 alpha and 2 epsilon chains, and then Portland 1 is going to be 2 zeta and 2 gamma chains. Now, once we get from the embryonic phase of development into the fetal phase of development, we then transition to hemoglobin F. And that is going to be made up of 2 alpha chains and 2 gamma chains. Now, I want to point out that from the point of hemoglobin F on into a normal adult hemoglobin, we always are going to have 2 alpha chains, and it's only going to be the second set of globin chains that differ. So if we have, for example, an alpha thalassemia with a complete deletion of alpha globin chains, we cannot actually have hemoglobin of any sort. And we're going to talk about that when we moving forward. But just a reminder that alpha chains are imperative. Without alpha chains, there are no types of hemoglobin. Okay, so hemoglobin F is a baby is born with lots of hemoglobin F or fetal hemoglobin, 2 alpha chains, 2 gamma chains, and then between the ages of 6 months and 1 year, that will transition into a more normal hemoglobin A. We always have a little bit of fetal hemoglobin though present, and there are some hemoglobinopathies in which higher percentages of fetal hemoglobin are present. And we'll talk more about those. I have this little red star next to hemoglobin A because this is our normal hemoglobin. It is 2 alpha and 2 beta chains, just like you learned about in biochemistry. And then we have hemoglobin A2, which is a variant, and this is going to be comprised of 2 alpha chains and 2 delta globin chains. Now, again, globin structure and production are governed by genetics, and the production of functional hemoglobin pairs 2 alpha globin chains with 2 beta chains together under normal circumstances, but we have thalassemias that are quantitative disorders of the globin chain synthesis. It can be alpha chains or beta chains, and usually it's one or the other. So we have alpha thalassemias, which are associated with deletions of alpha globin chains, or beta thalassemias, that are associated with the deletion of beta genes. So let's talk about those. I'm not really going to comment too much on this slide simply because I kind of went over it. But what I do want to say is that, again, this diminishes to very low levels as the baby gets older, but there's always going to be a very, very small amount that remains throughout childhood. Okay, so I kind of already talked about hemoglobin A and A2, but let's talk about hemoglobin, adult hemoglobin. So although adult hemoglobin is predominantly the A variety, so about 95 to 97, we do have a subtype of A2 that is also found in very small quantities, and that is normal. The abnormality would, you would think that it would be an abnormality when we have higher levels of A2 than are expected. And then we have that hemoglobin A2 that's made up of 2 alpha and 2 delta chains. Now, hemoglobin A, which is our normal adult hemoglobin, or I guess I should say our most predominant or our predominant hemoglobin, is produced in utero in very small concentrations. So embryonically and fetal development, you do see some hemoglobin A, but typically that hemoglobin F is the predominant hemoglobin that you would see in babies, and then converts to a high concentration of hemoglobin A 3 to 6 months after birth. I say 6 to 12 months based on the information that I understand, but it's just going to slowly decrease the fetal hemoglobin, and then you'll have higher concentrations of hemoglobin A. And then hemoglobin A2 production begins shortly after birth and continues at a slow rate throughout a person's life. And again, it's going to be very small quantity, 2 to 3%. We talked about variant forms of normal hemoglobin. Now, when I say normal hemoglobin, I don't know if normal is quite the right word, but let's say variant forms of hemoglobin. So this would be hemoglobin that started out as A2 and then binds with something other than oxygen, okay? So for example, carboxyhemoglobin is hemoglobin that has a higher affinity for carbon monoxide than oxygen does. Really what it is hemoglobin that has bound to carbon monoxide, and it does so 200 times more tightly than oxygen. And of course, this results in oxygen deprivation and tissue necrosis. It can lead to death. Many people have died of carbon monoxide poisoning. A lot of times they just get a headache to start out with, and then they go to sleep one night and never wake up. Sulfhemoglobin is the binding of hemoglobin to hydrogen sulfide, and that produces an irreversible change in that polypeptide chain. So this causes actual denaturization and precipitation of Heinz bodies, very much like some enzymatic deficiencies that are utilized to protect our red cells from oxidative purposes or oxidative processes, excuse me. A methemoglobin is hemoglobin with iron in the ferric state. Remember that 3+ state is no good. We want the ferrous state, which is a 2+ cation. So we've got we've got a problem when we are in the ferric state because that methemoglobin results in poor delivery of oxygen and cyanosis, which means someone may turn kind of blue. If you ever hear someone being cyanotic, that is, they kind of turn a blue color due to lack of oxygen and hypoxia. So that cyanosis can occur with methemoglobin levels of greater than 10% and hypoxia at greater than 60%. So once we have a problem with oxygenation, we start to see that discoloration in person's skin and lips, for example, their mucous membranes, specifically under the nail beds, all of those things. I just want to make a small point that I forgot to mention. So sulfhemoglobin can result from different types of drug ingestion. We know that there are some sulfa-based antibiotics that used to be really prevalently used, and some of them still are, but that can cause or sulfhemoglobin to occur. But with the removal of the sulfa drugs or the, exposure, um, the red cells should go back to normal. However, again, the ones that have been affected are irreversibly changed. So one more recap on this hemoglobin function. Of course, we know that hemoglobin's main function is to carry oxygen throughout the body. Let's talk about our 2,3-BPG or DPG. Those names are interchangeable. It is a compound that in which hemoglobin relies upon to enable our hemoglobin molecules to offload or pop off the oxygen molecules that are bound to our heme groups, okay? So when we have, I'm going to look at this picture here, look at my pointer, we have our 2 alpha chains, our 2 beta chains, and each one has a heme group. And right here in our oxyhemoglobin, we have 4 molecules of oxygen bound to our hemoglobin. And when the oxygen becomes depleted, so this has a high affinity, means it likes oxygen and it's holding on to it. This would be either in a normal resting basal respiration or maybe even a left shift. But when there's an increased need for oxygen, this 2,3-BPG will come in here, brace itself between the 2 beta globin chains, and cause a conformational change or shape change in the beta globin genes that causes the oxygen to be popped off of all of the all of the hemoglobin. So this is going to indicate a decreased affinity for oxygen. Those beta chains kind of pull together once the DPG or the 2,3-BPG is here, and it will then, after it's released the oxygen, it'll then come together and expel that 2,3-BPG. So in circulation, it can go back, pick up more oxygen, and then another 2,3-BPG will insert itself in between the beta globin chains and pop it off again. So depending on the need. And remember, that's going to be caused by a decrease or hypoxia occurring in the circulation. Okay, so here's just a reminder of what we discussed in our cellular respiration. Just remember that at about 40 millimeters of mercury, the hemoglobin molecule is still at about 75% saturated, and that is in the instance of our normal basal metabolic rate. That is going to shift to either the right or to the left based on our circumstances, our environmental factors, and those factors are going to influence the affinity and the release of oxygen. So in the condition of a right shift, or in a condition of hypoxia or decreased or insufficient oxygen, the oxygen dissociation curve is going to shift to the right. So we're looking at this red line now. We are first going to see an increase in those 2,3-BPG or DPG levels in the red blood cells. So that means they're going to wedge themselves between those 2 beta globin chains. That is going to cause a decreased or a lower affinity for oxygen, which means they're going to release the oxygen that is bound to the hemoglobin molecules within the red cells, which will then result in more oxygen being released to the tissues around. That means instead of just the normal 25% of oxygen released, we are going to have greater than 25% of oxygen released. Because remember, in our normal basal metabolic rate, only about 25% of the oxygen that is bound to our hemoglobin is released per one full circulation of the body circulatory system. So it could go around 4 times before all of the oxygen is completely depleted from the cells that we have in the body in circulation. Now, remember that 2,3-BPG is going to hop onto the red cell, wedge itself in between the two beta chains so that essentially the oxygen will pop off and go into the tissues. Then the beta chains, once they release the oxygen, will kind of contract, pop off that 2,3-BPG, go back around through the lungs, pick up more oxygen, and the cycle will continue until the hypoxia has been resolved. And then we will return to our normal basal metabolic rate. Now, the opposite is true in the case of a left shift where we are going to hold on. There will be decreased 2,3-BPG. We are going to have a higher affinity and a lower release of O2. And like I said, with the left shift, the decreased levels of 2,3-BPG, hemoglobin has a higher affinity for oxygen, and less than 25% of the O2 is released to the tissues. And again, this is one of the reasons why people who drown in very cold temperatures have a higher survival rate and can maintain, they can maintain less degree of tissue death due to hypoxia because those 2,3-BPG levels are decreased. The hemoglobin holds on to the oxygen, and so it can make more circulations because less than 25% of that O2 is released to the tissues simply because there's less demand for it. Even if the patient becomes hypoxic, the cold temperatures decreases their basal metabolic need. And so a lot of patients who have to be resuscitated, say they fell through the ice and they were pulled out and they've been under the water. You hear these crazy stories of someone being under freezing cold water for 20 minutes. Well, if that was warm water, that patient would probably not come out unscathed. They probably would not be able to survive that. But in the case of cold water, because of the decreased metabolic rate and that left shift, oxygen is maintained in circulation for a longer period of time. So really interesting. And in order to, if a patient does pass away, they can't call time of death until that patient has been brought up to body temperature, because a lot of times hypothermia is the cause of that patient's non-response rather than the drowning. If that makes sense. I hope that made sense to you. Okay. All right, so these are some of the dissociation curve conditions that you should be aware of. The shift to the right, we're going to see a decrease in pH, so that means the patient will become more acidic in terms of their blood pH. There will be an increase of CO2, an increase in temperatures can affect that, and then an increase of 2,3-BPG or DPG. And the opposite is true with the shift to the left, an increase in the pH, so the patient will become more alkalinic, a decrease in the CO2, decrease in temperature, and decrease in 2,3-BPG. All right, catabolism of hemoglobin. So again, as the erythrocyte ages, the following processes typically occur. Again, we know the membrane becomes less flexible due to decreased production of ATP, calcium buildup, the concentration of cellular hemoglobin increases, Enzyme activity, particularly glycolysis, diminishes, which is another cause of decreased ATP production. And all of those ion pumps are no longer able to work appropriately. So, yeah, it's um, just a little crazy. So, reviewing that senescent RBC process, those are taken out of circulation by the reticular endothelial system via extravascular catabolism. Macrophages, primarily in the liver, primarily in the spleen, some in the liver, phagocytize those red blood cells. And normally about 90% of red blood cell destruction is extravascular, meaning it takes place outside of the vessels. Iron is transported in the plasma by transferrin, that is our transporter protein for iron, to be recycled by the red bone marrow in the manufacturing of new hemoglobin. The globin is then catabolized in the liver into its constituent amino acids and then enters the circulating amino acid pools for recycling. And while we know that about 90% of red blood cell destruction due to senescence occurs extravascularly, we do have about 10% that occurs intravascularly. And this intravascular destruction is an alternate pathway to erythrocyte breakdown. So this process normally accounts for less than 10% or about 10% of erythrocytic destruction. And this is in the instance when we see free hemoglobin released directly into the bloodstream. We see dissociated alpha and beta dimers that are picked up by haptoglobin, and then haptoglobin and the hemoglobin complex get carried to the liver for further processing. It can be a very bad situation in the process if it's occurring in excess, and haptoglobin picks up the free hemoglobin. But once the haptoglobin has been all bound up by free hemoglobin that is circulating, we then have a spillover into the urine. This is the basis of many of our hemolytic anemias, such as transfusion reactions when, for example, a patient receives blood that that is not compatible with their genotype. So, yeah, I mean, obviously, intravascular catabolism is much, much worse than extravascular catabolism. In the case of senescence, most of that is extravascular. In the case of hemolytic processes, that is more likely to occur as an intravascular catabolism. So when we have intravascular red blood cell breakdown, we have free hemoglobin, which binds to free haptoglobin or haptoglobin. We have alpha and beta dimers. So these alpha and beta dimers are filtered through the kidneys. And then we have haptoglobin hemoglobin complex is sent to the liver. But in the case that we have an overabundance of free hemoglobin and not enough haptoglobin to bind and pick it up, we actually will then have spillover of hemoglobin into the urine, okay? The renal tubular uptake capacity is approximately 5 grams per day of filtered hemoglobin. But beyond this level, hemoglobin appears in the urine, so we'll be able to actually see it, and we call that hemoglobinuria. Not always. Sometimes hemoglobinuria is sometimes just picked up chemically on our dipstick, but sometimes you can see it. And then while in the urinary tract, hemoglobin gets oxidized into two pigments. So we have oxyhemoglobin, which occurs in alkaline urine, and then methemoglobin, which is in acidic urine. Hemoglobin that isn't processed by the kidneys nor bound to haptoglobin becomes methemoglobin in circulation. And we have a carrier protein called hemopexin that binds to that methemoglobin. And then that complex will bind to albumin to form methemalbumin. So we'll have like a triplex. I don't expect you guys to know all of that. I do want you to know what carrier protein is as a backup to haptoglobin. I want you to know about hemoglobinuria. I just, want you to know the general overall process that happens. I don't expect you to know every single detail or the biochemical mechanisms that are occurring, but I would like you to know what hemoglobinuria is, what happens when all of our haptoglobin is bound up, what the carrier for the for the hemoglobin in circulation is once our haptoglobin is bound up. All right, so that's it. This isn't so important to memorize, but it is important for your FYI. Um, this was a recap. I know it's repetitive, but repeating is always helpful in memorizing and learning. So thank you for joining me today. We will continue on with a new topic. We'll be moving on to anemias pretty soon, where we'll get more information about some of the things that I touched bases on today.